Okay, the other half of the adaptive immune response. We just talked about the cellular response with T cells. Now we'll talk with the humoral response, which will concern B cells. If you recall from the first figure in the book, figure 12.2, this is where we went through the 4 steps. We're now going to talk about them in a little bit more detail. So we'll talk about presentation, activation, proliferation, and differentiation in this video. And I will talk about antigen elimination, step 4, in another video. B cells are not as complicated in their presentation. As a matter of fact, B cells don't actually need to have a reaction with a T cell or an APC. As you can see in figure 12.18, what we have are T-independent antigens. There are BRCs, remember those B cell reception proteins, BCRs, B cell receptors, I always get those backwards. And the BCR that is on the antigen, you can see the antibody that's sticking out, they can go ahead and bind directly with an antigen. As a matter of fact, you can see in that picture that the epitope of several places on that pathogen have bound with several BCRs on our B cell. This will automatically activate our B cells. When they get activated in this manner, they can go straight to proliferation, which is, just like in T cells, mitosis. We undergo mitosis, we create many cells, and then they will undergo differentiation. And for B cells, the 2 different cells they will differentiate into are plasma cells, which have the antibodies. These are your effector cells. And then they will also differentiate into memory cells. However, you can also have T-dependent antigens. So you recall in our T cells, we had those CH12 helper cells here. Remember, the CH2 helper cells are what can activate the humoral response. This is one way that the cellular and the humoral response bind together. So we can see in figure 12.17, a B cell in this scenario will still come in contact with an antigen. So B cell is going to phagocytize an antigen. So we can see that in the first part. As it brings it in, it will break it down, or sometimes it might even keep it whole as they show in the picture, but usually it breaks it down. And then you'll have an MHC2 that will then bind with the antigen. That will then migrate over into the plasma membrane of the B cell, just like it would do for T's. So this will then migrate to the plasma membrane. And then from there, it will have a coupled response with the Th2 receptor. This is where it can interact with our Th2, our helper cell 2 cells. At this point, this is where you can see we will then get the releasing of cytokines. And if you recall from figure 12.15, this is when you will release interleukin 2 and 4 in order to get activation of the B cell. So our T-dependent antigens is a much more complicated scenario. You have to go through almost the same process you did as a T cell. But once it gets activation, that will then start proliferation, i.e. mitosis, will develop more cells, and differentiation into either plasma cells or memory cells. Both activations will lead to the same result. So there are 2 different pathways B cells can take to become activated.